Okay, so here's a short video to accompany today's lesson, because I can't be there, about genetic diversity, some of the ways in which we check the diversity in a population, and also then, if you like, the real grand story of life and what happens to that genetic diversity. So in front of us we have 3 of exactly the same species of iris. But you can see that there are definitely, I was going to say definitively, there are definitely 3 collections of genes that code slightly differently for the same things. So at the bottom we have a pure yellow iris, in the middle we have a white iris, and then behind we have a sort of gentle sort of mauve iris. They're all the same. So if pollinating insects were to go from one to another, the pollen of the yellow would successfully reproduce to make seeds if it was mixed with the ova of the white. So this tells us that whilst there is genetic diversity, there's not enough. You have to apologise for the traffic. Everyone's going to work. There's not enough difference for them to be a different species. Okay, so we've got 3 different alleles to work with. Okay, we understand that. Those alleles can be mixed individually in the plant by meiosis, and then when the pollen of another plant comes along, you get your random fertilisation, and you can get new varieties. So there can be, within these irises, mixing of those alleles. So, as a gardener, I could make a new variety that might be a mix between the yellow and the violet, or the yellow and the mauve, and I might be able to get something lots of other gardeners would really love. But in the wild, of course, it's all about attracting insects. Now, if they all attract insects equally, and so therefore their pollination is just as successful, and the colour of those petals doesn't affect anything else about the life of the plant, what you'd expect is you'd expect that none of those alleles come to dominate. So what that means is you'd expect the frequency of a particular allele to be the same as the frequency of another allele. So especially when it doesn't really change the success of a plant, the frequencies stay quite similar. And in actual fact, if I zoom out, you'll see that, roughly speaking, in all the irises, you've got a reasonable distribution. So the allele frequency of these 3 within the small population of my front garden is the same. And that indicates that maybe the mutation that produces colour to the petals doesn't really impact on the success. Okay, so they're all the same species. The actual genetic diversity is dictated by mutation and meiosis, but then the prevalence or the frequency of a mutation is when we start to introduce some form of selection. And so that's what we're going to just look at briefly today. It's quite a light lesson, and then tomorrow we're going to talk about, okay, what do we call, how do we label some of those selections. The importance of this genetic diversity is, for example, this iris is stronger as a species because it has 3 different versions of its petal colours. So if some mutation happened in the insect that pollinates it most frequently, say for instance, their eye receptor cells couldn't see the mauve one as successfully, so therefore didn't recognise it as a flower, they'd stop visiting the mauve one. So therefore that allele has become less successful, so it will not have the same opportunity to breed. And what you'd find is that then the white and the yellow versions of the gene get pollinated more, and their allele frequency comes to dominate. But it's actually the diversity that gives strength. And that leads on to what I want to do on Wednesday, which is just talk about why ecology and understanding the world around, the natural world around you in terms of the plants and the animals, actually equates so importantly for humans. And that absolute key is there is strength in diversity, because this, just in the colour of their petals, can cope with 3 dramatic changes. Okay, and now you see it in much more wild types. So I'm going to show you another one. Here is a foxglove, and this is the classic allele for the foxglove, which is these beautiful markings thought to attract bumblebees. They are their general pollinator, but in actual fact there are alleles that give you very different colour morphs. Okay, now as soon as gardeners get involved, we'll see tomorrow, as soon as gardeners get involved, one form of selection can happen very, very quickly. Now this one is interesting because actually not only is it a different colour morph, but actually, I don't know if we'll be able to see, actually it's got something else that's different. I don't know if you can see that on the video, but it's also, this one's very hairy. Now, does that help or hinder? We don't know. Don't know enough about foxgloves. But those three morphs again, they show that there are different alleles. Those alleles have been formed by mutation, mixed by meiosis, and now they are in the population. They can be selected for or against. So genetic diversity builds in strength to a species because it makes it more able to cope with different versions of the environment. Many of them are not as easy as flowers, many of them you can't see because there could be something different about the amount of water these irises need. One could have a larger surface area on its roots because of an allele, and the environment could change. I could get really bad at watering, and the one that survives drought the best will be the one that survives, changing the frequency of that genetic diversity. Okay, so just to recap, genetic diversity itself is caused through mutation and changes of DNA, which is then amplified or worked with by mixing of alleles in sexually reproducing organisms, okay, which then with random fertilization mixes it again. Genetic diversity doesn't necessarily offer greater success in the moment, but since the only time genes are transferred in the life of an organism is when it reproduces, when it produces offspring. If the individual's genetic makeup means they can have more babies, then the alleles it contains can stay in the population. So within a population, allele frequency is the number of times that allele is present. And as I said, if an allele say there's 5 alleles and their allele frequency is 20%, it kind of hints that that allele is not being selected for or against because it's staying the same. If, however, you had 5 alleles and you found that actually 92% of the population was one allele and then 2% was the other 4, making 100%, you always have to account for the whole population, it indicates that that 90% is currently the best for the environment the organism finds itself in. However, by having the other 3-4 alleles, you are building in genetic strength, because over time at each point of reproduction it could be that the environment changes and one of the other 4 becomes the better one. So that is genetic diversity. How we measure it is the frequency of the alleles in the population. Okay, and then if you add in time, you can change the allele frequency depending on what is the most successful route. There is strength over time in diversity. Have a lovely day, everyone.